Hi, thank you for being here today. I'll be talking about the role of RNAscope in spatial mapping of gene expression in tumors. For those of you who are not familiar with our technology, RNAscope is an in situ hybridization technique that was specifically designed to overcome some of the traditional limitations of an ISH assay while still providing very high specificity and sensitivity of detection. The RNAscope technology is the perfect solution for spatial analysis of your target gene expression. It can help interrogate complex tissues like the tumor, where you have different cell types with varying degree of gene expression. The three major parts of our technology are the target probe design, the signal amplification strategy, and the detection and quantification system. Because of our unique probe design and amplification strategy, it is the perfect solution for analyzing RNA biomarkers and potential therapeutic targets. ACD will design a specific target probe against the gene of your interest. Our unique signal amplification strategy ensures very high signal-to-noise ratio, and our detection and quantification system allows for visualization of a single molecule of RNA at single cell resolution. Our assay can be applied for detection of mRNAs, long non-coding RNAs, splice variants, highly homologous sequences, short targets, and point mutations. And our assay can be applied in FFPE tissues, fresh and fixed frozen tissues, PBMCs, or cultured cells. So this gives researchers ample of flexibility to use this technology in different sample types and for detection of different targets. In this next slide, I'm going to go in a little bit more detail to talk about our probe design strategy and the signal amplification system, the two key features of this technology that make it stand apart from some of the techniques that are out there. As you can see on your screen, I have depicted the RNAscope probe as a Z molecule, and this is mainly to highlight the three different regions of the probe: the pre-amplifier binding site, the linker region, and the target binding site. As the name suggests, the target binding site is the area of the probe that will bind on to your target sequence. When we design the probe, we design it such that two Z probes have to bind in tandem next to each other on your target sequence. Now, this forms the foundation of the pre-amplifier binding site, to which pre-amplifier molecule comes and binds to the Z pair. Followed by this, there's the binding of amplifier, and then you have your labeled probes binding on to the amplifiers. This signal detection strategy allows for very high sensitivity, and we can detect even the most low expressing targets. Now, as I mentioned previously, for the pre-amplifier to bind on to the RNAscope probes, the two Z molecules or probes have to be bound in tandem. If a Z probe binds non-specifically to another mRNA molecule, then the pre-amplifier will not bind to a single Z probe, and there won't be any amplification tree formed. As a result, no signal will be detected. Now, this ensures very high specificity of detection, giving you a high signal-to-noise ratio. In addition to our RNAscope assays, we also have a system called the Periscope assay, which is specifically used for detection of targets that are short, between 50 to 300 nucleotides in length. Now, these include your exon junctions, point mutations, highly homologous sequences, etc. The probe design strategy between our RNAscope and Periscope assays also varies to some extent. For our RNAscope assays, we design a pooled probe of 20 ZZ pairs, and for our Periscope assays, we have a pooled probe of 1 to 3 ZZ pairs, mainly because we are targeting a shorter sequence. In this slide, I have elaborated the breadth of our product portfolio. We offer assays that can allow single-plex detection, duplex detection, and up to 12-plex of target detection for RNAscope, where you can visualize 12 targets simultaneously. For our Barescope assay, currently, we can detect up to 2 targets simultaneously. We offer both the RNAscope and Barescope assays in a manual platform and an automated platform. Currently, we have over 26,000 target probes in our catalog. But if you're interested in a probe or a target chain that is not currently in our catalog, we can design a custom probe in less than 2 weeks. We are very excited to announce that in the next couple of months, we will be launching our microRNAscope assay, which is specifically designed to detect microRNAs and ASOs and siRNAs. Now, because of the flexibility of our assay, we are able to use RNAscope for a multitude of applications and in different research areas. As you can see on your screen, this technology has been used for detection of long non-coding RNAs, CDR3 regions on TCRs, GPCRs, point mutations, for validation of high throughput data, and so on. And because of this, it has been used in basic science research, for neuroscience and developmental biology, in infectious disease research, for understanding emerging pathogens and studying inflammation induced by infections, for biopharma discovery and development research in cell and gene therapy analysis to assess the preclinical safety and efficacy of these therapies, and finally, for cancer clinical research, wherein it has been used extensively in understanding the tumor biology and studying different immune cell markers in immuno-oncology. For the purpose of today's presentation, I'll be focusing on the applications of RNAscope in oncology and immuno-oncology. In this slide, I have summarized the different applications of how RNAscope and Barescope assays have been used in cancer research. They have been used to visualize tumor heterogeneity, study the varying degree of expression of a specific target in different parts of the tumor, to characterize immune cells and soluble factors, visualize a specific subpopulation of immune cells, and study the expression of cytokines and chemokines within those cells. Then to specifically detect TCRs, to identify specific CDR3 regions and identify clonal T cells, to detect splice variants, which can be pretty significant in cancer research, as certain splice variants are known to promote cancer growth and metastasis. Next is to identify non-coding RNAs, again, an important family of regulatory RNAs that are non-coding but can influence cancer growth and progression. To spatially map sequencing data, which includes validation of high throughput data like single cell RNA sequencing and visualizing and validating those data in a morphological context. And finally, to detect engineered immune cells, such as CAR T cell therapies. Let's start with the first application, which is to visualize tumor heterogeneity. In this particular example, we have used the RNAscope assay to visualize PD-L1 mRNA in lung cancer tumors. On your screen, you can see 4 different panels depicting expression of PD-L1 in 4 different lung cancer tumors. And you can appreciate the difference in the degree of expression of PD-L1 in these tumors. Although they are the same type of tumor, the expression of PD-L1 varies significantly between these 4 samples, highlighting the heterogeneous expression of PD-L1 in lung cancer tumors. In this next image, we have shown the expression of PD-L1 in the same tumor, in different parts of the same tumor, where you can see that PD-L1 is expressed at a much higher degree in the top right corner compared to the bottom left corner, showing that PD-L1 not only exhibits inter-tumor heterogeneity, but also shows intra-tumor heterogeneity, where its expression can vary depending on the specific niche within the tumor. And this can be extremely significant for targets like PD-L1, or CTLA-4, which are important targets for immunotherapies. Next, I want to show you an example of how we were able to compare the differential expression of some of these immune targets between two lung cancer tumors. From our TMA core of lung tumors, we are focusing on core number 2E2 and comparing it to core number 2F1. As you can see on the screen, we have used our duplex assay to visualize different immune checkpoint markers, including PD-1, PD-L1, PD-L2, LAG3, TIM3, and CTLA-4. It is very obvious that the expression of all of these immune checkpoint markers is significantly higher in the core 2E2 compared to the core 2F1. And this is also evident from the quantification detailed in the table below. When we compared the expression of immune cell markers within these two cores, we see that the 2E2 core, also called as the hot core, shows a much higher degree of expression of these immune cell markers compared to 2F1. The markers we have chosen specifically include markers like interferon gamma and CD8, which are markers for cytotoxic T lymphocytes, FOXP3 CD4, which are markers for regulatory T cells, CXCL10 CD68 for M1 macrophages, and CCL22 CD163 as markers for M2 macrophages. And it is clearly visible that the degree of infiltration of these immune cells is much higher in the hot core compared to the cold core, potentially suggesting that a patient with the 2E2 tumor was more likely to have a successful immunotherapeutic response compared to the patient in the 2F1 core. So using this technique, we were able to visualize the immune cell infiltration between two different tumor types and understand potential response to therapy looking at these markers. And RNAscope also helps identify the cellular source of these secreted cytokines and chemokines. Here's an example of how researchers have used the RNAscope assay to visualize the heterogeneity of Wnt ligand expression in these pancreatic tumors. They have developed a pancreatic tumor organoid library and then studied the expression of Wnt ligands within these tumors to understand the dependency of these tumors on the Wnt signaling pathway. And what they observed was, compared to the normal pancreas, the W negative tumors, or the tumors that were Wnt non-secreting, expressed significantly lower levels of these three Wnt ligands compared to the W positive or Wnt secreting tumor organoids seen in the rightmost column of this panel. So using this technology, they were able to understand the expression signature of Wnt ligands within these different pancreatic tumors and study the dependency of these tumors on the Wnt signaling pathway, giving them novel insights on the tumorigenesis of pancreatic cancer. Now we are going to move on to the next application of RNAscope, which is to use RNAscope to characterize immune cells and study soluble factors. One of the most important workflows of using RNAscope is its ability to combine RNAscope with immunohistochemistry or immunofluorescence. Because of the natural complementarity of these two technologies, we can use that in combination and visualize RNA and protein simultaneously on the same section. Now, this becomes extremely important in immuno-oncology, wherein you can use specific antibodies to mark certain immune cells and then study the gene expression using RNAscope within those specific populations of immune cells. And here is an example of how we've utilized this joint ISH-IHC workflow. In the cervical cancer, we used the CD3 antibody to mark all the T cells and then study the expression of PD-L1, PD-1, and interferon gamma using RNAscope. So combining our multiplex assay with immunofluorescence, we were able to identify a specific cell type and then study gene expression within those cells. Using RNAscope, we were able to highlight the cells that were expressing high degrees of interferon gamma, potentially identifying the active T cells within the cervical cancer tumor microenvironment. And this particular image also lets you appreciate the single molecule and single cell resolution that you can get with RNAscope. A single punctate dot here refers to a single transcript or molecule of RNA, and you can visualize these at the single cell resolution. This joint workflow of using RNAscope with IHC or IS is not only limited to the fluorescent platform, but can be done on the chromogenic platform as well. In this example, we have visualized CD68, CXCL9, and IL-12 in the lung cancer tumors. Using the CD68 antibody, we have potentially marked all the macrophages and then studied the expression of IL-12 and CXCL9, which are both soluble factors, using RNAscope. So this allows us to pinpoint or identify the cells that are secreting these soluble factors within this lung cancer tumor. A similar example of using CD4 antibody in combination with TGF-beta and FOXP3 probes. And here you can nicely see how we've been able to visualize the CD4 positive T cells in the stroma of this lung cancer tumor, and then co-localize the expression of TGF-beta and FOXP3 to these cells. And so this tells you that using RNAscope, you can study gene expression in different compartments of the tumor. Now, this is an example of how researchers have used this dual ISH-IHC or dual ISH-IF workflow for their research. This paper, published in Nature last year, utilized the RNAscope duplex and multiplex assays to study the effect of LIF, or leukemia inhibitory factor, in promoting pancreatic cancer growth. As you can see here, they have compared the expression of LIF and keratin 19 between normal and cancer tumors. And you can see that keratin 19 has been used to mark the tumor cells, and LIF is expressed predominantly in the stroma, suggesting that LIF is not expressed by the tumor cells, but by a population of cells called the pancreatic stellate cells, which are present in the stroma and are secreting LIF. So any potential effect that LIF has on the cancer cells is a paracrine effect and not an autocrine effect. They further went on to show that when they co-localized LIF PTPRC with keratin 19 antibody, they were again able to demark the tumor cells and then show co-localization of LIF with PTPRC, which is CD45, emphasizing that LIF was expressed in the stroma and not in the tumor cells. They also went on to show that LIF is definitely a factor promoting pancreatic growth in this particular study, because they saw that circulating levels of LIF were significantly higher in cancer patients compared to normal samples. Similarly, they showed that there was a strong correlation between LIF mRNA and decreased disease-free survival. One of the next applications I want to talk to you about is a very exciting application, which is using base scope assays to detect CDR3 lesions on TCRs. In the last decade or so, there has been a lot of emphasis on engineered T-cell therapies. And to do this, it's important to identify specific clonal populations of T cells. And using our base scope assay, we can design a specific probe against the CDR3 lesions of TCRs and identify clonal populations of T cells. Because these are really short hypervariable regions, it can be very challenging to detect these with a morphological context. So our Periscope assay being a specialized assay for short targets, we are able to specifically design probes against these CDR3 lesions and identify clonal populations of T cells. Now here is an example where we designed CDR3 probes against the alpha and beta chain of JERCAT CDR3 and then CDR3 alpha and beta of TCRs. So when we looked at the expression of these CDR3s in the particular cell line, we were only able to detect positive signal in that particular cell line, highlighting the specificity of our Periscope CDR3 probes in detecting target of interest. And this was again nicely demonstrated in this paper in Nature Medicine from Dr. Balco's lab at Vanderbilt, where they were treating a patient with anti-PD-1 therapy, and the patient was responding well to the therapy initially, but later showed adverse event to this therapy, which caused encephalitis, which was ultimately fatal. They wanted to understand what kind of immune response was induced by this therapy that led to encephalitis. So upon doing TCR sequencing of this tissue, they were able to identify a specific sequence that was predominantly expressed in this tissue. They designed a Periscope probe against this sequence, and when they co-localized this with either CD4 or CD8 antibody, they were able to see that this TCR sequence co-localized with CD4 positive T cells and not with CD8 positive T cells, thus showing that this population of T cells that induced encephalitis was a CD4 positive T cell population. Upon further analysis, they were able to identify that it was indeed a CD4 positive memory T cell population that was causing this encephalitis. And using the Periscope assay, they were actually able to visualize this population of T cells within this encephalitis tissue. Here's another example of how RNAscope was used to study T cells. In this case, these researchers were studying the gamma delta T cells and their role in potentiating tumor growth. Now, gamma delta T cells are ideally known to act against tumor growth, infection, and tissue damage. But with age, the role of gamma delta T cells in preventing tumor growth is altered. And what these researchers are trying to show is that compared to younger mice, in the aged mice, you see that the gamma delta T cells show increased expression of inhibitory cytokines like IL-7 and IL-17, which in turn potentiate tumor growth. So again, using our multiplex assay, they were able to visualize the soluble factors like IL-7 in combination with TCRs, specific to gamma delta T cells. Next, I want to talk to you about another very exciting application of the RNAscope technology, which is to identify and detect engineered immune cells. So in the last decade or so, there has been a lot of research around developing CAR T cells for specific antigens in solid tumors. This is a study in collaboration with Bluebird Bio, where they were trying to develop CAR T cells against BCMA and ROR1 antigens. And using the RNAscope technology, we were able to design a specific probe that was designed against the 3' UTR of the CAR vector that went into these CAR T cells. In addition, we were also able to visualize markers like granzyme B, interferon gamma, which are markers for active T cells or engaged T cells. So for the anti-BCMA therapy, when they designed these CAR T cells and injected them in the preclinical mouse model, they looked at the expression of these CAR T cells in different tissues. Here, they're looking at the expression of the BCMA antigen and the CAR T cells in the mouse spleen. On your right-hand side panel, you can see that the spleen tissue of this mouse has no BCMA antigen expression. And on your left-hand side, you can see in red the CAR T cells that are present in the spleen. But you do not see any green signal, which would be indicative of positive granzyme B expression. So in this normal mouse spleen, although the CAR T cells are present, they are not engaged because there is no BCMA antigen present. On the other hand, when we are looking at the xenograft tumor of this preclinical mouse model, the tumor now has the BCMA antigen present. And these CAR T cells are not only expressed in this particular tumor, but they're also showing engagement indicated by strong expression of granzyme B signal. So this shows you that in the presence of the specific antigen, the CAR T cells are able to engage and detect the antigen. So this shows you that this anti-BCMA CAR T cell therapy was effective and specific and was not showing any on-target of tumor effects. On the other hand, when they looked at the efficacy of the anti-ROR1 CAR T therapy, the result was something different. They were seeing a lot of toxicity in the mouse lung, and they were trying to identify the reason behind this toxicity. When they used IHC to identify the expression of ROR1, they saw that they were not able to pick up any ROR1 signal in the lung tissue, but they were still getting this pulmonary toxicity because of CAR T therapy. But when they used RNAscope to visualize ROR1 expression, as you can see, there is very low but some basal level of ROR1 expression in this lung tissue, as a result of which these CAR T cells were not only present but now engaging within the lung, causing pulmonary toxicity in this particular preclinical model. And this showed them that the ROR1 CAR T therapy had off-tumor, on-target, but off-tumor effects, which can be potentially toxic. Similar to lung, they also observe hepatic toxicity, but when they actually looked at the expression of ROR1 antigen and CAR T cells in the xenograft tumor, they found that for some reason these CAR T cells were now not present at that higher degree in the xenograft and were not engaging either, potentially because they were exhausted. So ultimately, they deduced that the anti-BCMA CAR T therapy was successful, was effective, and did not show any non-specific effects, whereas the anti-ROR1 CAR T therapy showed some on-target off-tumor effects and was therefore potentially toxic. And here is just an example of how, using the multiplex assay in the BCMA, anti-BCMA model, we were able to now visualize the CAR T cells indicated in green, all T cells indicated in white by CD3 antibody, engaged T cells indicated in red by granzyme signal, and expression of interferon gamma indicated in pink. So we were able to distinguish between CAR T and endogenous T cells and then visualize which T cells were engaging. Next, I want to talk to you about a very popular application of the RNAscope and Basescope technology, which is to spatially map sequencing data at single cell resolution. In the last few years, single cell RNA sequencing has gained a lot of importance, and this is because it is a technique that allows visualization of gene expression at the single cell level. And although you don't get the morphological context, you get information of each and every cell within the tissue. And this allows you to study the different cell populations within the tissue and understand their specific functions. But what you lack is the morphological and spatial context that you get by RNAscope. So a lot of the times, researchers will do the single cell sequencing analysis and then come back to RNAscope to validate and visualize their data and spatially map using morphological context. So here's an example that was published last year from Harvard and MIT collaboration, where they analyzed glioblastoma tumors using single-cell RNA sequencing and identified 4 different cellular states in which glioblastoma cells exist. Now, these different cellular states were also characterized by expression of specific molecular markers. what they wanted to do was visualize these different molecular and cellular subtypes in situ. And they used the RNAscope duplex assay to pick specific markers pertaining to each and individual cellular state and visualize these cells within the tissue. But what they discovered was the glioblastoma cells can not only exist as a specific cellular state, but they can exist in a hybrid state where they exhibit characteristics of more than one cellular state. And they observed this by showing co-localization of markers specific to different cellular states in the same cells using RNAscope. So using our technology, they were able to visualize this hybrid state of tumor cells in glioblastoma, which can potentially add to our understanding of how this tumor works and how this tumor progresses to help identify better therapeutic strategies to treat this deadly malignancy. Similar to the example in the previous slide, the same group of researchers also published another paper where they did single cell analysis on medulloblastoma tumors and identified different phenotypic subtypes of medulloblastoma cells based on their Wnt signature. And they categorized these as 4 different Wnt molecular subgroups. Using the Periscope assay, they were able to visualize these different subgroups by co-localizing the markers within the tumor tissue. Here's another example where these researchers characterized cancer-associated fibroblasts in this breast cancer tissue using single-cell sequencing analysis. They were able to identify 4 different populations of CAS, and using RNAscope, they were able to visualize these different populations within the tumor tissue and validate their single-cell RNA sequencing data. So next, I want to dive deeper into studying how the Periscope assay can help advance your oncology research. Detection of splice variants can be very tricky because these are products of the same gene and can structurally be pretty similar, although can have different functions. So detecting splice variants at the protein level can often be challenging. But using our specific Periscope probes, we can distinguish between the different splice variants of a single gene. Now here's an example of how these researchers use the Periscope assay to identify the delta 16 HER2 splice variant in breast cancer and other tumors. So the probe design strategy that they employed was they designed a probe that spanned exon 15 and exon 17 of the HER2 transcript. This would allow for detection of only the transcripts that had exon 16 missing. That way, they could detect the delta 16 splice variant. They also designed probes that span exon 15 and exon 16 that would now detect the wild type HER2, and then a third probe that would span exon 17 and exon 18, which would detect both the wild type and the splice variant. And they used these 3 probes to study the expression of the HER2 receptor in breast cancer, colorectal cancer, and gastric cancer tumors. So here they saw that in these samples that are mentioned on the screen, they were able to see the expression of the delta 16 HER2 splice variant, the wild type HER2, as well as they saw positive signals for the probe, which spanned both the splice variant and the wild type HER2. But with the HER2 antibody, they were not able to distinguish between these different splice variants and wild type. So they used the Periscope assay to stratify patients for Herceptin therapy, because patients that have the delta 16 HER2 splice variant respond really well to Herceptin treatment. And using our Periscope assay, they were able to identify patients who would respond better to this therapy and stratify other patients who would not benefit from this particular treatment. So the specificity of our assay allows for specific detection of splice variants that are significant in promoting tumor growth, like the delta 16 splice variant. Now, here's an example where the researchers used the Bioscope assay to look at the circular splice variant of androgen receptor and its role in prostate cancer progression. The circular form of this androgen receptor is known to be associated with progression and metastasis of prostate cancer. On the slide, you can see the comparison between the expression of this circ-AR splice variant of androgen receptor between hormone-naive and castration-resistant tumors in this particular example. You can see that the expression of the circular RNA is significantly higher in the castration-resistant tumors compared to the hormone-naive tumors, indicating that these tumors that are more resistant to any therapeutic intervention are showing increased expression of the splice variant, potentially suggesting that it might have a role in driving the growth and progression of this tumor. So this indicates that our Bioscope assay can not only be used for detection of linear RNA, but can also be used for detection of circular RNA splice variants. Another application of the RNAscope technology that is less common is detection of long non-coding RNAs, but these can be extremely significant when it comes to oncology. Long non-coding RNAs can contribute to the initiation and progression of several malignancies. Since they don't have any protein products, detecting relevant long non-coding RNAs has been quite challenging. Using specific target probes, we can identify long non-coding RNAs in any tissue and any tumors. Here's an example of how researchers have used RNAscope to visualize a newly identified long non-coding RNA called nuclear leucab within the nucleus of these lung cancer cells. They established that this nuclear long non-coding RNA was upregulated under hypoxic conditions and promoted an aggressive phenotype within the lung cancer tumors. Another group used the RNAscope technology to demonstrate the upregulation of the long non-coding RNAs in metastatic breast cancer. About 90% of the brain mets were positive for this particular target. What I would like you to appreciate here in the first example is the subcellular resolution that you can get with RNAscope if you have a really good microscope. You can we have co-localized nucleic nuclear RNA with NEAT1 nuclear long non-coding RNA using our multiplex fluorescence assay. So you can visualize both these targets within the nucleus. In addition to both these examples, we have a lot more publications that have used RNAscope to visualize the long non-coding RNAs of interest in cancer cells and tissues. This is another example of how we can use different probes to identify long non-coding RNAs that are significant in a particular malignancy. Here we have used 4 different probes in 4 different tumors, and you can appreciate the different pattern of expression of each of these long non-coding RNAs, wherein the Gas5 long non-coding RNAs expressed in breast cancer shows both nuclear and cytoplasmic expression, whereas the MALAT1 long non-coding RNA shows specifically nuclear expression in this lung cancer tumor. So with our technology, you can get that subcellular localization of targets like these long non-coding RNAs, which can be significant in understanding their function in tumor growth and progression. So I want to take the remainder of the time to talk about a very exciting collaboration between ACD Biotechnique and Nanostring Technologies, where we have combined our technologies in a single workflow to give you highly plexed spatial analysis with morphological context. RNAscope is now a part of the GeoMx DSP workflow, where RNAscope can be used as a molecular guide to direct the DSP, the GeoMx DSP on specific areas of the tumor, which can then be profiled using the DSP, quantified using the nCounter, and then targets of interest can be validated using RNAscope on a serial section. So this is a workflow which we believe can help address several questions for immuno-oncology research. And this is especially significant for samples with high heterogeneity. We wanted to show that this particular workflow can be made applicable for an actual study within a lung cancer tumor sample. So we've decided to investigate the immune cell infiltration and the expression of associated cytokines in the tumor microenvironment. So the first step is to identify regions of interest using specific RNAscope probes. The second step, as I mentioned, is to perform the digital spatial profiling using the GeoMx on these identified regions of interest. And then the third step is to validate or confirm the data that you get from your GeoMx DSP, again with that single cell and single molecule resolution with RNAscope. So to show the applicability of this technology, we decided to use the CD3 and CD19 RNAscope probes to interrogate this lung cancer tumor sample, and saw that different regions of this lung cancer tumor had varying degree of CD19 and CD3 expression. Some regions had higher degree of CD19 expression, while some regions had high degree of CD3 expression. And this indicates the differential expression or differential levels of immune cell infiltration within this lung cancer tumor. So the 24 regions of interest that were identified were a mix of high CD3, high CD19, and moderate expression of both targets. After identifying these 24 regions of interest, they were then analyzed using the GeoMx DSP. The GeoMx DSP panel that we chose is the IO panel, which consists of 78 immuno-oncology related targets. And what we saw was that there was a significant differential expression between the genes that were analyzed between CD19-rich ROIs and compared to CD3-rich ROIs, wherein the targets that were upregulated in the CD19-rich ROIs were mainly antigen-presenting cell targets, targets like CD40, CD74, whereas the targets that were upregulated and enriched in the CD3-rich regions of interest were mainly T-cell markers and chemokines and cytokines. Now, CD19-rich regions we expect are regions that are high in B cells, and CD3-rich regions are regions that we expect are high in T cells. So after having analyzed this by digital spatial profiling, our next step was to validate these some of the key targets that we analyzed using RNAscope on a serial section. So for that purpose, we selected ROI6 and ROI11. As you can see, ROI6 is a CD19-rich ROI, and ROI11 is a CD3-rich ROI. So this way, we can compare the differential gene expression between these two ROIs that we got from GeoMx DSP analysis, and then pick certain key targets that were differentially expressed and come back to RNAscope to validate their expression on a serial section. So the key targets that we picked for validation are targets like CD8, interferon gamma, CXCL9, and CCL5, which are key players in the cancer immunity cycle. And these were differentially expressed between ROI6 and ROI11. So using our multiplex V2 assay, we were able to visualize these targets and compare their expression between the CD3-rich ROI and the CD19-rich ROI. As you can see in this image, the expression of all of these targets was significantly higher in the ROI11 compared to ROI6. Now, ROI11 is the ROI that is CD3-rich or T cell rich. And ROI6 is the ROI which is CD19 rich or B cell rich. So it was no surprise that these targets, which are mainly expressed on T cells, were expressed at a much higher level in ROI11 compared to ROI6. And it was nice to see that our expression pattern was concordant with the DSP data that you can see in this graph, showing that these two technologies are compatible and their expression patterns correlate with one another. With RNAscope, as I mentioned earlier, you can get that single molecule and single cell resolution. So after having identified your key targets from your DSP data, you can come back and visualize your key target expression with morphological and tissue context. So with this joint workflow, we really wanted to address all the questions in spatial genomics, where we've used RNAscope as a molecular guide to identify key regions within a heterogeneous tumor. Then, using the genomics DSP panels, we can profile these selected regions of interest for target gene expression, in this case, targets related to IO, and then visualize the key targets that are differentially expressed using RNAscope on a serial section. So this way, we're hoping to provide a complete solution for spatial genomics, combining the strengths of both these technologies to provide highly plex spatial profiling with single cell and morphological resolution. Our technology has been widely applied over the last few years. As you can see, we have over 2,700 publications as of last month, and we have been used in various different research areas. But cancer and neuroscience are top research areas for publication. In addition to our broad applications in cancer and neuroscience, we've also widely been applied in infectious disease research. And in the current environment, it's imperative that we expedite our research for COVID-19 and help identify therapeutic and vaccine strategies that will help control this pandemic. And we are trying to contribute to this by having probes for COVID-19 that would specifically identify this virus, and also have probes that would identify the host cell receptors, chemokines, cytokines, immune cells, and finally, pathological and physiological markers to study tissue homeostasis. To learn more, do not hesitate to reach out to our support. Finally, we have a lot of material related to oncology and immuno-oncology. As you can see on the screen, we have white papers and app notes, and we are also going to be presenting 4 different posters at the AACR meeting later this month. So please reach out to us with any questions. You can reach out to either of the email addresses. And in the current environment, we know a lot of you cannot get to your lab, but you still have research and grant deadlines to meet. So at ACD, we have a dedicated group of researchers who are safely working in our office to help advance your research. We have pharma assay services and academic assay services for pharma and academic customers, where you only have to send your samples to us. We'll do the RNAscope analysis for you, do the data analysis quantification, and give the data back to you in 4 weeks. So if you want to learn more about these services that we offer, do reach out to us and contact our support. Thank you.